As with all of our publications, this paper was compiled and written by a task force of volunteer scientists and other subject matter experts. The CAS liaison for this paper is Dr. Anna Dilger. Anna is an associate professor at the Department of Animal Sciences at the University of Illinois, Urbana-Champaign, where she teaches courses in animal growth and development, meat science, and contemporary issues. Anna's research program focuses on the efficiency of meat production by understanding fundamental regulation of muscle and fat growth, as well as improving the quality and sustainability of meat products. She is a member of the editorial board for both the American Journal of Animal Science and the Meat Science and Muscle Biology. Anna, thank you for presenting today, and I'm going to turn it over to you at this point. Thank you.
There we go. I think I'm unmuted now. And so before I get started with an overview of this particular paper, I want to take just a second to recognize the members of the task force who served as the authors of this commentary. And so they include Dustin Bowler from the University of Illinois, Min-Ho Kim and Jess Krieger from Kent State University, Jennifer Martin from Colorado State, Andy Milkowski from the University of Wisconsin-Madison, Paul Mozdziak from North Carolina State, and Brian Sylvester from the firm of Foley, Lardner. We'd also like to express our appreciation to our reviewers for this particular article. And with that, I will start our overview. And so, like many of you, those of us at CAST have long been interested in and curious about this new technology to produce food from cells cultured from animals. But whenever we tried to get our arms around this particular issue, it seemed to change because it's a really rapidly evolving field and technology. And so for this particular commentary, instead of trying to put together a be-all, end-all paper that explains everything about these technologies that would surely be out of date by the time that we publish it, instead, we decided to focus on those challenges and uncertainties regarding this technology, particularly in the areas of the techniques, the sale and regulation of these products. And so for today, I'm going to address a handful of questions that the task force identified and a little bit about where that technology and the issue is today. And so those questions include what techniques will be used and are being used to make these products, what types of products are expected to be available, who will be responsible for regulating those products and how do they plan to do that, how will these products be labeled, and what food safety risks may exist with these types of products.
Why I don't do this?
And so starting first with the techniques to produce these eventual products, we can kind of break this into a workflow that includes cell line development, cell cultivation, the structuring of tissue, and then tissue cultivation. And so starting with cell line development, in these types of products, cells serve as our base of product. And there are many different sources for these cells. They might include biopsies of living animals, cells derived from embryonic stem cells, or other adult stem cells that exist in animals' bodies. Currently, the cells that are being targeted are mainly muscle and adipose tissue, or fat cells, because these are the main cell types that are present in traditionally produced conventional meat. But in order to eventually culture whole tissues, we may need to isolate and cultivate other types of cells that are also present in meat, things like connective tissue or blood vessels. And in order for these cells to be developed and to grow for use in products, in culture, these cells must grow, divide, and then mature into kind of mature and functional cells. And so once suitable cell lines are developed and have been screened and targeted, we need to then be able to culture them at a large scale. And so this means moving from very well-established benchtop techniques that have been developed over really the last 100 years of kind of developmental biology research. Most of the time, those cultivations happen in maybe some milliliters or even a few liters at a time. But really what we're talking about is moving to thousands or tens of thousands of liters and out of a culture dish and into a bioreactor. And so this will require a very large volume of cell culture media. And it is that media that contains the nutrients that are needed to kind of feed those cells as they grow, divide, and mature. But that media also contains the growth factors and hormones that are needed to direct that growth and division and maturation or differentiation of the cells. And so the cultivation of cells in bioreactors is really only the first potential step in this process. And there are several companies that have or are rapidly attaining this stage of kind of cell cultivation and production. And this alone can lead to food products to come to the market. But really the ultimate goal of this idea of cultivating tissues to produce food is to completely mimic the texture, the taste, and the composition of traditional meat. And this will require culturing several types of cells at once as tissues, which is a very complex arrangement of cells that mimic living tissues in animals. And so theoretically, these cells can be seeded into matrices of either plant or animal origin and then cultivated together as a tissue. However, this is going to require the development of several new technologies to really attain this goal. And so when we think about the technical challenges that need to be overcome in order to really scale up and bring these cultivated cell products to market, I've summarized some of those here. The first is the identification and characterization of suitable cell lines. These cell lines will need to be screened for ease of propagation, their eventual nutritional content, traits of palatability, or ease of manufacturing into meat products. We also will need to work to lower manufacturing costs and scale up our production. And so when we think about manufacturing cost, that has to do with the components of the media and the ability to produce a large scale of media, and then also the energy cost of bioreactors. And scaling up has its own challenges, is that what may work in a culture dish or a few liters does not always translate into thousands of liters in a bioreactor. We will also need to develop waste streams for used media or other waste of these processes such that they can be safely disposed of, recycled, or reused. And then we will need to see the development of some allied industries, like those who would make media, and then the training of a skilled workforce. And so that second question that we wanted to address is, what do these products look like? What do we expect these products to be when they do become available? And we've divided that into 3 different phases of eventual production, with phase 1 being of the soonest to arrive at the market, and phase 3 being much later. And so those phase 1 products will likely be cultivated cells being mixed in to what we would currently consider a meat analog. And so that would be a plant or fungus type of meat product, mimicking a meat product, and using those cultivated cells to provide nutrients or flavor to that overall product. Our phase 2 products will be composed entirely of cultivated cells. But these products would likely mimic the current ground or formed products that we see on the market, like hamburgers, sausages, or nuggets. They wouldn't have that tissue structure that you would expect in a steak or a roast. Those would actually be our phase 3 products. These products would be cultivated tissues containing many different cell types and would ultimately mimic the taste and mouthfeel of steaks and chops and roasts and other whole muscle products. But as I said before, the technologies needed to develop those products still need to be invented. Probably one of the more sticky issues in terms of the production of food from cultured animal cells has to do with regulation. And who exactly will be responsible for regulating the products, and how will they do that? And in the last couple of years, we have seen a tremendous amount of organization and movement in this area of regulation. And so when we think about these eventual food products from cultivated animal cells, they will have aspects that will overlap and span the areas of responsibility currently covered by the FDA and by the USDA. When we think about the things that the FDA currently regulates that may be present in these food products, they are things like the ingredients used in meat and poultry products, veterinary products or reviewing new technologies, biologics and gene therapy products and other tissue products that may currently be made in bioreactors are all in the purview of the FDA. On the other hand, the USDA has responsibility for products that contain meat. And that includes the 100% inspection of all animals and carcasses during the slaughter process, pre-approval of labeling of these products, and the oversight of implementation of HACCP programs in order to mitigate food safety risks. And so in 2019, the USDA and FDA formalized a memorandum of understanding which outlined their planned cooperative approach towards the regulation of these food products. And they established a working group to address how they are going to integrate their proposed regulation, with the FDA responsible for cell collection and cell line development, and the process by which those cells and tissues would be propagated or cultivated. And then there would be a transition from the FDA to the USDA at the time that those cells and tissues were harvested, and the USDA would be responsible for overseeing and regulating the manufacture of food products. That working group currently has 3 areas that they are addressing. The first is the pre-market assessment, which is being led by the FDA. There's a group who is working on how exactly this transfer of jurisdiction will happen, and the USDA is taking the lead on labeling. Given that the USDA will have responsibility for the regulation and oversight of final products, it is the expectation that the USDA will extend HACCP plan and sanitation protocols similar to traditional meat production into these new products. That their regulation will include the inspection of products and records and the pre-approval of labeling. And the USDA has also been charged with developing additional requirements that might be needed. If there are new challenges in ensuring the safety and accuracy of labeling of these products, it is up to the USDA to develop those additional rules. If the regulation is a particular sticky issue, even more sticky is how exactly these products will be labeled. And this is a very large area of uncertainty because currently there are no products on the market that require labeling. But when we think about how meat products are currently labeled, they are currently defined by a series of standards of identity. And what these standards of identity list are things like the origin of the meats, the addition of other ingredients, types of processing that are used in the production of these meat products, and a little bit about their nutrient composition, especially the quantities of protein or fat that these products may contain. And so it's interesting to think about how maybe these definitions could be expanded and extended to products containing cultivated animal cells. In the commentary, we go through 3 different scenarios that are really, frankly, thought exercises to really think about each one of these phases of production, maybe the challenges that are produced when we start thinking about those products. And so I've highlighted that just a little bit here. When we think about something like a hamburger from a standard of identity standpoint consists entirely of beef with no additional ingredients and can contain no more than 30% fat. And so when we think about cultivated cells being incorporated into a product like that, would a product containing only cultivated cells from cattle be considered hamburger because those cells are beef? And would the USDA maybe require that the source of those cells be called out specifically, or because they come from cattle, would it currently fit the definition of hamburger? Another thing to think about is the concept of cooked ham. And so cooked hams are currently classified based on a protein fat-free system. that results in lots of different labels like ham and ham with natural juices and ham with water added. And so when we think about cultivating cells to maybe include in these products, would they need to be from ham muscles? Traditionally, ham comes from the back leg of pigs, and so would the source of those cells also need to come from the back leg? And could pig muscle cells be added to ham, and the protein from those muscle cells count towards protein fat-free, even if it wasn't a whole muscle that was being incorporated? Our last example here is breakfast sausage, which is a sausage prepared with fresh or frozen meat byproducts, and has a lot of different standards of identity, including that it can contain no more than 50% fat. And so how will regulators view protein and fat being derived from cultivated cells towards these nutritional standards? And when we think about calculating the concentration of restricted ingredients like nitrite and phosphates, will we take into account the weight of these cultivated cells, or will it be calculated without them? Finally, the last question that we wanted to address were, what were the food safety risks of these products? All foods for human consumptions must be evaluated for potential biological, chemical, or physical hazards to food safety. Put plainly, when new products come to the market, consumers need to be absolutely confident that their food is safe. And so anticipated food safety risks for products containing cultivated animals or tissues, when we think about it, they're a mixture of familiar threats, but also novel ones. Some people have hypothesized that cultivated cell products may actually be safer than traditional meat, because those cultivated cell products will lack a slaughter environment. And without that slaughter environment, will eliminate many of the food safety risks that are posed in that environment. But I think it's important to remember that these products will be at the same risk as traditional meat during processing, packaging, and harvesting for contamination by other organisms in the meat processing environment, very similar to traditional meat products. And so as a way to think about what are these food safety risks, we can kind of separate it into the different phases of production and think about what might happen and therefore what we need to guard against. When we're thinking about cell line development and cell banks, we do worry about contamination of those cells, either during collection or during propagation, such that we need to be confident that the cells we are cultivating are the cells that we want. We also worry about perhaps reagents being used in that cultivation process that are not currently rated as food safe. It may potentially leave residues in products. There's some similarity to the current regulations around veterinary products and food additives in traditional meat production, where we do go through safety evaluations to make sure that no residues are present which are harmful for people. In the cultivation of cells, it is possible to have contamination within bioreactors. And it's possible, when we think about it, that that risk is similar to the risk that animal diseases might pose at slaughter. And is the reason that inspectors inspect animals and carcasses during that slaughter process. And so how will we adapt that system to think about now contamination within bioreactors? But really it's that post-harvest environment, the manufacturing, processing, and packaging of meat that we come to are very familiar threats. Contamination of meat products during manufacturing, the presence of allergens, these are probably very similar to the current concerns that we have of safety during meat processing. And so with that, I am happy to open us up for questions. We are going to turn it back over to Kent in order to get organized just a little bit. We appreciate you joining us today, and I am happy on behalf of our task force to take any questions. Thank you.
Thank you, Hannah, for that great presentation. And thank you for sharing some of the highlights of this. We're receiving several questions, and we're going to be sharing those questions with you in just a few minutes. And as we prepare for that, I did want to cover just a couple of highlights of some things coming up. On May the 12th, we are going to be announcing the 2020 Broadcast Communication Award winner. And that will be done by a webinar. Dr. Frank Mitloner, the 2019 award winner, will be a presenter on that program. So please join us for that webinar. That will be at noon Central, 1 p.m. Eastern Time. And then on May 18th and 19th, we're going to be releasing a new paper on the 18th, followed by a webinar on the 19th. And this paper deals with the need and challenge for effective stewardship of new pest management technologies in agriculture. And Dr. David Shaw from Mississippi State University will be sharing that information with you. Next slide, please. As I shared with you at the beginning of the webinar, we are very pleased to have a number of experts join us today for our panel discussion, who will be available to answer questions. Please welcome Jess Krieger with Kent State University, Jennifer Martin with Colorado State University, Andrew Milkowski with University of Wisconsin-Madison, Paul Mozdziak with North Carolina State University, and Brian Sylvester with Foley and Lardner LLP in Washington, D.C. Panelists, thank you for your participation. And now I'm going to turn the program back over to Anna to lead the question and answer, the question period.
Thanks, Kent. We have a series of questions and really appreciate people with their curiosity typing in their questions. For those that we don't get to, we do plan to follow up, at least in written format, with some of the questions afterwards. And so to start off our questions, I'd like to start with Jennifer Martin. The question is, what are the steps that you would undertake if you were going to begin to design a food safety program for cultivated cell products?
Hi, Anna. That's a great question. I think in order to design an effective food safety program for these cultivated cell products, we would really start in the same place that we start with traditional meat products, and performing a risk assessment and assessing hazards that exist within the system, similarly to what we would do in a HACCP protocol. So before we can effectively implement a food safety system, we have to identify and assess risks that are associated with the food that we're producing. And this would be really no different from that. So I think that's a great starting point, would be to look from cell cultivation to finished product and identify risks, establish the critical control points for those risks, and then develop solutions to mitigate and minimize risks within the system itself.
Thanks, Jennifer. Our next question is for Jess. Jess, what are some of the current challenges to tissue cultivation? We talked about how that's an emerging technology that we don't precisely have down yet. And do we need to master those techniques before we see any products come to market? Juan, it looks like there, Jessica, you're ready to go.
Hey, sorry, what was the question?
So the question I had for you are what are some of the current challenges that we have with tissue cultivation? And do we need to overcome those challenges before we're going to see any of these products come to market?
Yes, definitely. So generating tissues, it's a complex process because tissue contains multiple cell types that have defined locations. An example of this is how skeletal muscle fibers in meat are arranged in these parallel bundles, and there's blood vessels that grow around them to provide nutrients and oxygen to the cells. So the main two types of equipment that are going to be necessary to grow meat in the form of tissue are bioprinters and bioreactors. Bioprinters are 3D printers, and they can print living cells into tissue in these specific patterns to recreate the structure of native tissue in the body. So special bioreactors need to be developed to serve essentially as life support systems for tissue. And these are called tissue manufacturing bioreactors. And they can supply nutrients to the cells within the tissues to keep them alive. Overall, bioprinters and bioreactors for tissue cultivation are still at a pretty early technology level. So there's lots of work that we're going to need to do to perfect these systems so we can actually use them for the cultivated meat industry. There's additionally some significant cost issues involved since these are expensive equipment types. So we have a long way to go before we can actually tissue engineer meat.
And so Jess, thank you for outlining all of those challenges, but do we need to overcome all of those challenges before we start seeing products come to the market?
Well, the good news is we don't. Developing a tissue product will help us make products that mimic cuts of meat, like steak or slices of bacon, but we won't necessarily see those products entering the market first, since, like I mentioned, there's a lot of work that needs to be done in the development of just the equipment that we would use to tissue engineer products. So currently, we can grow individual cells pretty easily, and we don't necessarily need to synthesize them together into tissue. So for example, cells contain the flavoring compounds that generate the taste of meat. So cells themselves can be used to flavor products. So I have fun news. Recently in my lab, we grew a cultivated burger where we added about 10 grams of cells to 90 grams of plant protein and spices. And it tasted like a burger that you would derive from animal meat. So we can essentially use cells themselves as a food additive without necessarily having to tissue engineer them into any special type of product. So I think the first kinds of products that we'll see on the market are going to be, using cells as, skeletal muscle cells and fat cells, meat cells, as food additives to plant-based burgers and nuggets and sausages and so on.
Thanks. The next question we have is for Andy. And so Andy, when we think currently about labeling meat products, could you explain a little bit about how currently we assess that labels are accurate in terms of added ingredients or ham, protein, fat-free specifications? And how can we extend that then to these eventually cultivated cell products?
Well, thank you, Anna. Well, currently, if you are a meat product processor, producer, and you want to sell a product across state lines and interstate commerce, you would have to submit to the USDA your formula and an outline of your manufacturing process prior to getting those products into commerce. And so there's a review process there that will enable you to, the USDA, to govern and make sure that those labels are accurate and legal in that regard. In terms of operations, that responsibility transfers to the in-plant inspector, who is, as part of his or her duties, reviewing production records to make sure that what is being made is consistent with what was approved in the original label application. And then with respect to your PFF question, going back to the slide that you had about grades of ham, that's a process control requirement where the finished ham being produced would be analyzed for moisture, protein, and fat, and a calculation to derive the PFF value would be done. And this would be performed on a lot-by-lot basis. And then the trends would be tracked via what's called a Q-sum statistical process control approach. And if you got out of into some type of a deviation, moving outside of the ham that you are currently labeling for, you would have to institute some sort of a process or formulation change, largely with respect to the amount of water you use in the process, to make sure you get back into specification. And again, the compliance with that is checked by the implant USDA inspector. As far as cultivated cells go, how they would fit in really needs to be determined by policy on how these are going to be considered in moving into different products, such as ham that we've been discussing. And so the first layer would be really determined by policy in the USDA and how they apply that to a particular situation. And then the rest would flow with implant inspection.
Thanks, Andy. The next question we have is for Paul. And so Paul, one of the issues that was raised in the commentary was the harm or the risk that might occur if we have contamination in our cell lines. And so what are some ways that we can protect cell line integrity and be sure that we are cultivating the cells that we want to?
Thanks, Anna. That's really an excellent question, and it's really, when you get down in the weeds, somewhat of a complicated question, because cell line integrity is really a major problem in phase 1 research labs and academic research labs. And back in 2012, it was one of the top problem to be solved in biology. It hasn't really been solved in academic research. The but is that on the other side of it, cell line integrity is not really a huge problem in the biopharmaceutical industry, where they work in a CGMP environment with very good documentation and very good cell line authentication methodologies. So, I think the points that I want to make is that, the greatest risk of contaminating multiple cell lines is when the process is open at isolation. One of the advantages of cell culture is that it's a closed process. And with it being a closed process, the opportunities to risk mixing one cell line with another cell line once they're established are relatively minimal. So the simplest answer is to simply work with one cell line at a time, to have that cell line quarantined as it goes through each process. And maybe the more complicated answer is just very simply to do DNA analysis on the cells that you're working with at the beginning of a process and at the end of the process. And what would play into that would be working with qualified cell banks. So, I think that gives a little bit of an overview in 3 minutes.
Thanks, Paul. So our next question is for Brian. And so the USDA and FDA have decided to work together in order to regulate these products. Are there other examples of that type of collaboration between federal agencies? And maybe what challenges do you see for this collaborative process?
Absolutely, that's a great question. So I think that we don't have to really look outside of the FDA, USDA to understand collaborative working arrangements because there's a lot of ongoing collaboration between USDA and FDA on a range of issues. If you look back to around 2007, 2008, there was cloned meat, and there the FDA and USDA partnered where the FDA looked at the safety of cloning meat and the USDA worked hand in hand in that effort, and FDA ultimately issued a risk assessment that USDA relied upon. Currently, when we're talking about ingredients that are used in food products, and meat products and poultry products in particular, FDA considers the safety aspects and USDA FSIS considers the suitability. So there's a lot of MOUs that help us in our everyday work when it comes to food regulation. And I think the good news here is that the MOU outlines the broad strokes of what FDA's role will be and what USDA's role will be, especially when we're talking about meat and poultry products that are subject to the Federal Meat Inspection Act and the Poultry Products Inspection Act. There's a lot of details that remain to be filled in, so that might be where I see some difficulty, but it's not insurmountable. So the pre-market interagency group that the FDA has currently, they're meeting with companies in this space, the startups in this space. They're learning about the technology, and that will inform any type of guidance that comes out to help shepherd these products to market. So right now, I think there's a lot of conversations going on, and that those conversations will inform the direction, the details of what the agency's approach will be, if that helps answer that question.
It does, thank you. And so circling back around then to Jess, there are lots of claims being made about the eventual sustainability of these products. And some claiming that the cultivated meat products or cultivated cell products might have improved sustainability compared to traditionally produced meat. I do not expect that right now you can tell us whether or not that's true, but how can we start to think about determining the sustainability of these processes, and maybe what are the things that need to be investigated to answer those questions?
Yeah, so that's a great question. Primarily when I think about sustainability, I think about the effects of climate change on the environment. And really what we're trying to do with the cultivated meat industry is to try to create a method of meat production that has less of an impact on the environment and climate change than we see in some of the other aspects of meat production with regards to industrialized factory farming. So, you know, one of the aspects there is, you know, the direct emissions that can come from the animal agriculture industry. So one strategy that we would like to use in our industry is reducing direct emissions as much as possible by using not only renewable energies, but doing away with the direct emissions that can come from animals. So by producing cells in these big cultivators, we can essentially bypass any methane emissions that come from cattle. So in this way, we think that the advantage for the cultivated meat industry is really going to be that we can create a system that is sustainable for the environment. So there is a lot of work that needs to be done there, but that's a target goal for our entire industry. And since we're using manufacturing equipment and we're developing new types of equipment that can use renewable energy resources, I think we're in a prime position to try to tackle those sustainability challenges.
Great, thanks, Jess. Is there anyone else on the panel that maybe would like to expand on the issue of sustainability? I'm going to say no. That's okay. So the next question I have is for Paul, and it really centers around costs involved in cultivating these cells. And so if you could talk a little bit, Paul, about what are the costly parts of cultivating these cells, and maybe what efforts are going on to lower those costs.
Thanks, Anna. You know, the one thing that is obvious and that everyone talks about in terms of the cost is the cost of the media used to cultivate the cells. You know, I saw several questions in the chat box about serum in the media, and that serum is classically used in animal cell culture, and that tends to be the most costly part of the culture. But what we replace the serum with is recombinant growth factors for the most part, and those are also very costly. So that there's a great deal of effort being put forth to find alternatives to growth factors to bring the media costs down. So those are a couple of points. The other points that are being looked at are things like recycling media in terms of the bioreactor operations to reduce cost. And then another issue is to design new and different types of bioreactors to result in a smaller amount of media per unit yield of tissue at the end of the process.
Thanks, Paul. The next question I have is one for Andy. And so there's a question in the chat box, and I think it is a popular one that people think about creatively labeling. And so I'm sure we have all encountered at the grocery store some interesting spellings of things like patties or links that would describe meat products that maybe don't meet standard of identity. Do you think that that may be one route that the industry takes and maybe doesn't try to apply the standards of identity for meat products, but instead comes up with their own names or own labels?
That's the 600-pound gorilla in the room in terms of where we will go in the future. I think if you compare this to historical paradigm shifts in the food industry and other industries, there are a variety of opinions based on whether there's some self-interest or not involved there. And I don't know how it will play out. Conventional agriculture and meat production is certainly going to fiercely defend their turf to maintain what they would consider the integrity of what they are currently doing. And that's something that has centuries of practice. You could go to Europe and look at the German purity laws or some of the laws in Italy with regard to some of their traditional products. How it would work out here is up for grabs right now. I think that's part of the collaborative effort between FDA and USDA is how do they work through that? How do they satisfy the need for innovation and progress and moving forward, yet protect some of the very strongly held traditional opinions about different types of foods and foodstuffs, and it'll be interesting to follow it. At this point, I don't have a clue as to how that's going to play out.
I agree, Andy. It's certainly going to be an interesting discussion between these new cultivated cell products and our traditional meat products. And so the next question I have, I'm going to direct towards Brian, and it is about the concept of whether these products may be considered halal or kosher, and whether you're aware of any outreach to religious communities and talking about how they might view these products.
All right, thanks for that question. Every conference that I attend that's focused on cell culture, cell cultivated meat, they typically have a breakout session where they are discussing whether these products can be called halal or kosher. I know that the Rabbinical Council has been looking into this, so the conversations are ongoing. I don't know that there's any kind of final decision made yet, but it's definitely an interesting one that is provoking lots of conversation in all the right places. And I wanted to just add on to the comments on labeling that, you know, every company in the space seems to be very interested in ensuring that their products are referred to as meat or poultry, etc. But there's also the desire to distinguish their products. And I think the question then becomes, is it something that USDA mandates in a new standard of identity, or is it a voluntary sort of additional disclaimer that the product labels would bear? We're seeing a lot of state legislation from Oklahoma to Missouri to many other states that are kind of defending the turf of conventional agriculture interests. And I think those ongoing conversations are influencing what the federal regulators at the USDA might be thinking. And we know that there is a working group that's focused on labeling headed by USDA, and that they are actively considering the issue. So we'll have to stay tuned.
Thanks, Brian. A question for Jennifer about maybe some food safety risks. One of the comments, a couple of the comments in the chat box has to do with elements in the media and how those themselves might pose a food safety risk or a residue in those meat products. And so I know maybe a little bit outside of your current realm of research, but could you speculate a little bit about how we might approach those?
Yeah, I think one of the, you know, as with any developmental process, we have to consider any and all risks. And there are certainly the potential for risks of residues from the reagents used, which, as you mentioned in the presentation, aren't currently food grade. And so there are certainly potential risks that may be associated with the elements used in the media for cell culture that would certainly have to be explored. And I think that's a protocol that we would follow for any food safety system in our current meat production chain to assess risks that exist within the system itself. And some of those may be ones we're familiar with and know well, and others we have to evolve as our production systems evolve. And this would be no different. So I think there's absolutely the chance that they may pose a risk. And we would have to measure that risk, and then once we've assessed the risk, then we can implement food safety or strategies and potentially policy to help alleviate the risk that it may pose to a consumer that we're just not currently aware of. So I think definitely the potential is there, and we would approach that potential risk the same way we would approach a potential risk in our current food system.
Great, thank you. So Jess, a question for you regarding exotic meats. And so there was a question in the chat box about whether or not cultivated cell products are a way to perhaps satisfy some desires of people for some exotic meats and maybe cut down on the illegal or harmful poaching of those animals.
I love this question. Yes, this is definitely an opportunity for developing cultivated meat products that are not just, you know, standard in the diets of Americans or other communities around the world. We can select animals, basically any kind of animal that we want, and collect cells from them and develop their cells into cell lines, edible cell lines. So, you know, we don't have an ability to try lion meat typically in the United States, but if we can take a sample of skeletal muscle cells from a lion, there's no reason why we can't create a lion steak or a lion burger. Something that I found particularly interesting is there's some researchers around the world that are trying to resurrect the woolly mammoth, so to speak, by taking some cells that have survived from frozen woolly mammoths, and they've actually been able to culture them in a laboratory setting. So I've always thought it's a really cool idea if someday we could try different kinds of meat that aren't necessarily available to us in the current ecosystem that we have here on Earth. So maybe one day we'll have a mammoth beef burger.
And with that, and as we're all now envisioning the strange and exotic meats that maybe one day we will get to try, I would like to thank our panel for participating today. And with that, I will turn it back to Kent.
Thank you, Anna, and thank you to the panelists. Those were some great answers and great questions. I will share with you that if you submitted a question that was not answered, we are collecting those and we will work with the panelists, and we hope to post a document that contains those questions as well as the answers from our panelists on the questions that were answered today, as well as some of the questions that we weren't able to get to today. Just a couple things before we close. One, a reminder on the May 12th, Borlaug Cass Communication Award webinar. Please join us for that one. That will, again, will be at noon Central, 1 o'clock p.m. And then on the 18th and 19th, our next CAS paper coming out on the need and challenge for effective stewardship of crop protection technologies. The paper will be available on the 18th, and we will have a webinar on the 19th to summarize the highlights, key points, and hopefully have a panel discussion much like today's. Again, thank you for joining us today. I do want to acknowledge and thank our friends at Dairy Management Inc. who help us by providing this web platform and also the technical assistance that they provide in helping us do this. Thank you all for joining, and again, reminder, this webinar was being recorded and will be available on the CAS website later. Have a great day. Thank you.